Can I welcome you all and begin by reminding you about mobile phones. Please switch off. All of us, in varying degrees, have a major interest in the subject of today's lecture, aging. And I can think of no better person to induct us into the mysteries of the biological mechanisms of aging than Linda Partridge, showing how the mechanisms of ordinary aging have been conserved across evolution, and from the study of simple organisms one can extrapolate to what happens in humans. Linda Partridge is Weldon Professor of Biometry and Director of the Institute of Healthy Aging at University College, but she's very much European in that in 2008 she became founding director of the Max Planck Institute for the Biology of Aging in Cologne, and divides her time between Cologne and UCL and other parts of Europe too. And we're extremely privileged she's been able to come here and talk to us today in this schedule, which reflects her impressively active scientific life and the contribution she's made to the subject, which have brought her many distinctions, which I won't rehearse in detail. I know you'll join with me, and that Raymond and Beverly Sackler would also wish to join with us in welcoming Dame Linda Partridge to give the 19th Raymond and Beverly Sackler Distinguished Lecture on the New Science of Aging. Linda.
Thank you very much, Patrick, for an extremely kind introduction. And of course, it's an enormous pleasure and honour to be here and also to see so many old friends. So, as the title of the lecture suggests, ageing is a research area that's changing quite fast at the moment as a result of some quite key discoveries in recent years. And I hope during the course of this lecture to convey some of the excitement that currently pervades the area. Of course, our own ageing and mortality has fascinated not only ourselves, but scientists and philosophers ever since people have started to think about it. And none more than Robert Boyle, one of the very early founders of the Royal Society, who in the 1660s compiled a scientific wish list, which was a list of the things that he thought were most in need of a scientific solution, but were likely to present the greatest challenge. And at the very top was the prolongation of life and the recovery of youth. And although he didn't live long enough to see it himself, the prolongation of life has actually been happening very steadily in developed countries since the middle of the 19th century. It's illustrated here, the trend from the work of two demographers, and what they've done is simply to plot life expectancy at birth against the year in which the births took place for the country that was the world leader at the time in terms of life expectancy. So to start with, this was the Scandinavian countries, latterly it's been Japan. And you can see this extraordinary linear increase. It's gone on steadily at about 2.5 years per decade, 6 hours a day, since it started. And the horizontal lines and the points on the left simply represent various people's or organizations' predictions of when the trend would top out. But actually, the main point that this paper was making is that at the moment there's no sign of an approaching wall of death. It's not clear what the intrinsic limit on human lifespan is going to turn out to be. Although, of course, current trends in obesity and diabetes in the young may mean that we're about to see a generation where this slows down or even halts. And of course, that's led to some remarkable lifespans. I guess many of you are familiar with this lady. She's the official world record holder for lifespan, a French woman who died in 1997 at the age of about 122.5. And she actually met Van Gogh in her father's shop. He used to go there to buy paint. And I think it's interesting to say, well, why her? And two features I think are of particular note here. One is that she came from a very long-lived family. And human life expectancy or human lifespan is about 25% heritable, although that heritability seems to be concentrated in a few very long-lived families. But on the other hand, she wasn't a very good advertisement for a healthy lifestyle. She enjoyed her drink, and she did eventually give up smoking at the age of 119. So who knows what her secret was? But obviously this whole trend has to be celebrated as one of the crowning glories of human civilization because of improvements in lifestyle and healthcare. People are staying healthy as they get older, and so they're living longer. But of course it's coming with a very mixed press because it has a clear downside, which is that age is the major risk factor for the predominant killer and chronic diseases. So these are just compiled from various EU and UK statistical sources, and you can see the more or less exponential increase with age for these three. And of course, that means that there's a great burden of ill health falling on the older section of the population, which is causing considerable personal hardship and also economic difficulties. And those don't just apply to sufferers. The ageing population in the UK has been rated as unaffordable because of a storm of public debt combining with an increasing proportion of older people in the population. So the pressures on the biomedical scientists to come up with ways of trying to improve health during aging by tackling the underlying aging process itself. But until quite recently, the prospects for doing that have looked poor. And one of the main reasons is simply the complexity of the aging process. So this is a pictorial representation of a table that was produced some years ago by a pathologist, George Martin, in which he listed the various things that can go wrong in various human tissues during aging. And what it presents is a picture of great complexity of different things happening within tissues and between them. It almost looks as though there is no single underlying aging process that you could capture to ameliorate all or many of these simultaneously. Rather, what you're seeing is each tissue system accumulating damage in accordance with the kinds of insults of daily living that it acquires. And of course, that would carry a pessimistic message. It would mean that we would be looking at best at piecemeal investigation and intervention into the things that go wrong during aging. And very much by the same token, I think people tended to assume that the same would apply to these creatures, the laboratory model organisms. They're, of course, the workhorses of modern biomedicine. Very often, if we want to understand a process like gene expression or genetic transmission or metabolism, we start with a simple organism, a bacterium or single-celled yeast, then go to the multicellular invertebrates, and only when it's solved there, start to tackle the more complex mammalian situation. And that works beautifully because of the very powerful principle of evolutionary conservation. We can often take a gene from a human or from one of these organisms and put it in another, and it works perfectly well in the new context. But I think for a long time people thought, well, why should these organisms encounter the same kinds of problems as they get old? They live for very different lengths of time, their metabolism is very different, the mouse is warm-blooded, food supply is quite different. Why should there be commonalities in their aging process? And eventually, somebody who's my personal hero in this whole story stopped thinking too hard about aging and just did the experiment. What he did was to take the nematode worm, Caenorhabditis elegans, and ask if he could use a chemical mutagen to induce mutations that would make the worms live longer. And he found that he could. So his very significant contribution was to discover these mutant genes that meant that the median and maximum lifespan of the worm was considerably increased. And it was actually a long time. He discovered them in 1983. It was only starting in 1996 that the genes were cloned and sequenced. And they turned out to be part of a worm insulin-like growth factor signaling pathway. So this is a survival curve for worms that are mutant in DAF2, which is the single worm insulin IGF receptor. There was another mutant that became known as AGE1, which turned out to be in the catalytic subunit of the PI3 kinase. And it also turned out that a third gene called DAF16 in the worm was known to be required for these two mutations to extend lifespan. And that turned out to encode a forkhead BOXO, a FOXO transcription factor. So of course we now know that the way that this pathway works is that if we knock down the upstream activity in the pathway, the forkhead transcription factor becomes dephosphorylated, it enters the nucleus, and it can participate in control of gene expression. But I think the main interest of this at the time that it was discovered was that perhaps it might be conserved in other organisms. And eventually it became possible to ask that question in the creature where I do most of my own work, which is the fruit fly, Drosophila, because the pathway turned up there, not because of anything to do with aging, but because it controls growth and cell proliferation during development in the fly. So, of course, a lot of work was done on that. Some nice mutations and other genetic reagents were produced. And eventually our lab and Mark Tater's lab at Brown University simply rounded up some of them, put them on a standard genetic background and asked, well, do they extend lifespan in the fly, as they do in the worm? And it turned out that they did. So this is the gene that we worked on. It's a gene called Chico intracellular. It encodes the single fly insulin receptor substrate, which is an adapter protein that sits just below the receptor and helps transduce the insulin signal into the cell. And you can see the effect on growth. This is a Chico female on the right who's lost both copies of the Chico gene. You can see she's a dwarf. Her survival curve's in red on the right, and you can see that she's nice and long-lived. So are the heterozygous flies. They're almost as long-lived as the homozygotes, but they're completely wild type in body size. So the dwarfing and the extension of lifespan are not causally connected with each other. They're just two things that the pathway does. And in fact, we can take these flies through to adulthood and only turn on this expression of the insulin pathway once they're fully grown and still see the extension of lifespan. So again, the interest here was perhaps it would turn out that this pathway was important in mammalian aging. And starting in 2003, papers began coming out about mice. So the first one was a knockout of the insulin receptor in the white adipose tissue of the mouse, the Furco mouse. That was reported by the Kahn lab to be long-lived. And the Holzenberger lab found that if they made the mouse ubiquitously heterozygous for a null mutation in the IGF-1 receptor, they also got a long-lived mouse. I'm going to show you one that was produced by our consortium in London, partly because we did it, partly also because it's actually a Chico mouse. So this was done in the lab of Dominic Weathers, one of my long-term collaborators in London, led by Colin Salmon, who's now actually got his own lab in Aberdeen. And this is a mouse that's lost both copies of the insulin receptor substrate 1. There are 4 in the mice. And you can see this nice extension of lifespan in the females. One of the things that's interesting about this pathway is that when we knock down its activity, both in mice and in flies, we tend to see larger extensions of lifespan in females than in males. We don't know why that is at the moment, but it's rather a consistent effect. But one important thing about this study was that they did a very thorough phenotyping of the mice as they went through the aging process. And they found basically what we weren't expecting to see. So they've lost their insulin receptor substrate, and they actually start out life when they're young, rather insulin resistant. It looks as though they might tip over into diabetes as they get older. But the beta cells compensate and make up for the insulin resistance. And actually, as they go through middle and old age, their glucose homeostasis is very much better than that of the control mice. They also have a better immune profile, more naive T cells. Their motor function's improved. So you can test that by putting the mouse on a rotating rod, seeing how good it is at hanging on. They maintain youthful levels of function on that much better. They have a delayed onset of osteoporosis. These 2 mice here are sisters. They're littermates at the age of about 800 days. And you can see the cataracts on the control mice. Also, about 40% of this strain, as they go through aging, get this ulcerative dermatitis at some point. You can see it on the head and nape here. These mice are completely protected from it. They never get ulcerative dermatitis. So what this has given us is a result of a mutation in a single gene. Is this very broad spectrum improvement in health during aging involving systems that seemingly don't have any connection with each other. And that's, to me, why this is interesting. Of course, we would very much like to know whether any of this applies to humans. And at the moment, the main way that that's being investigated is by looking at genetic variants in human genes orthologous to the ones that have turned up in the animal studies, and looking at association with survival to advanced ages, 90 or 100, in human population studies. People tend not to do genome-wide studies on this simply because the population sizes that are being looked at are too small. There's not the power there. They're going in with candidate loci. And it's very early days, but some interesting things are turning up, particularly in this FOXO3A. So this is one of the 4 human orthologs of that gene that I mentioned in the worm, DAF16, that encodes the transcription factor essential for lifespan extension by reduced insulin signaling. We've recently shown that the fly ortholog of that gene is also required for extension of lifespan by reduced insulin signaling. And I think 7 independent studies now found that genetic variants in FOXO3A are associated with survival to advanced ages. FOXO1A has also turned up once. This is an interesting study. This is the Einstein centenarian study based in New York. They found that a variant in the IGF-1 receptor was enriched in their centenarians. And they actually went on in this study here to do some functional tests with it. So the actual consequence of the genetic variants in this 4K transcription factor, as far as I know, is still not understood. But this one, clearly, if you put it into mammalian cells, reduces the activity of the receptor, which is what you would expect. I've included this lady here just to make a point about centenarians generally. So in terms of their lifestyle, they're very average. So they're no less likely to smoke, they're no less likely to be overweight, and they're no more likely to take exercise. They seem to be very like the general population in lifestyle. And also people are starting to look now at whether they're any more likely to have a lower load of risk alleles for aging-related disease. So, of course, there have been a lot of GWAS studies now looking at or searching for risk alleles for particular disorders during aging. And it turns out that centenarians, again, are very average for that. They don't have a lower load of those kinds of alleles. So something else seems to be going on in these individuals that survive to great ages. Now, of course, what the insulin-IGF pathway does is to sense the nutritional status of the animal and to match costly activities, so growth, reproduction, wound repair, metabolism, to the nutrient status. And almost certainly what this network does, and the other nutrient sensing networks with which it's associated, are to mediate the effects of an intervention that's been known about for much longer, which is dietary restriction. So this was first discovered in rats, actually, and then subsequently in mice in the 1930s. So if you simply force the animal to eat about half of what it would take in voluntarily, you see a very large extension of lifespan. And because it's been known about for such a long time in these creatures, they've been very thoroughly phenotyped. And they are, to some extent, protected from almost everything that goes wrong during aging, including metabolic disease, neurodegeneration, and cancer. There are very clear benefits there. The two problems that they seem to have are with wound healing and with certain types of viral infection. They definitely do worse on those. But the aging-related component is greatly ameliorated. And since it was first discovered, various model and non-model organisms have been shown to have a similar response. If you reduce the amount that they eat, they live longer and generally maintain function better. And very recently, a study from the National Institute of Aging in the US on rhesus monkeys has come out. And there again, there's an increase in lifespan. They can't be restricted quite as severely as the rodents. They can only be pushed down to about 70% of voluntary intake. But then they're starting to show a very, very broad spectrum improvement in health during aging. There's a rash of papers coming out on these monkeys at the moment, but they look very like the dietarily restricted rodents. We'll probably never know if dietary restriction extends human lifespan because the effort of willpower involved is simply not compatible with doing a randomized controlled trial. Most people just cannot do it. In fact, some people do dietarily restrict themselves and also offer themselves to be studied. This is one such individual. Most of them are male for reasons that I think nobody quite understands. And they often turn up at aging conferences, and one of the most obvious things about them is that they don't look very happy. It's difficult. And obviously the measurements that we can make on them are considerably less than in the monkeys or the rodents, but many of the same things are turning up. So this particular paper looked at 12 biomarkers for risk of atherosclerosis and found that they were all down after 7 years of DR. And there are a number of other such studies coming out. But at this point, I'd like to start a little advertisement process for the fruit fly, Drosophila, because it can be used to address a lot of important points in this area, I think. And one of them is the actual nutritional basis of the response to dietary restriction. So flies do dietary restriction quite nicely. So what we do is to dilute this gelled food medium that many of you will have seen that the flies eat. And it usually consists of sugar, usually sucrose, yeast, sometimes some other ingredients. And if we dilute the food, for some reason the flies don't compensate by eating more of it. So if we give them dilute food, they get less to eat. And what happens if we do that is, first of all, lifespan increases to a peak under dietary restriction and then falls off a cliff through starvation. And the number of eggs that the females lay goes down steadily. And this is very typical of dietary restriction in all the organisms in which it's done. Yeast and worms show these responses as well, and the mice show the right-hand side. And it's often been assumed that this reduction in fecundity that tends to accompany the response to dietary restriction is somehow necessary for lifespan extension. And it's also often assumed that calories are what's important. You may have noticed in the previous slide that both of them say calorie or caloric restriction. And that's based on some work that was done many years ago with mice, in which it was suggested that it doesn't matter which macronutrient you use, whether it's the protein, the carbohydrate, or the lipid component of the diet that's used to take out the calories. What matters is the reduction of calories. And we already know that that's certainly not true in flies. We use a sugar yeast medium, and just by chance it turns out that sugar and yeast have almost exactly the same calorific value per unit weight. And if you dilute the sugar in the food, the flies essentially show no response. Neither their fecundity nor their lifespan responds to sugar dilution. All of the action is in the yeast component of the diet. So clearly there's a response to some specific nutrient or nutrients going on. So some years ago now, we decided to investigate exactly what was going on here. It was done by a graduate student in the lab, Richard Grandison, and Matt Piper, who was then a postdoc and is now again running his own lab at UCL. And what they did very simply was to measure the nutrients that are present in our yeast and then take flies that are dietarily restricted, so that on dilute food they live long, they don't lay very many eggs, and we simply give them back the components of the yeast and ask what responds, the fecundity or the lifespan, to different nutrients. And the first thing that turned up very simply was that all of the action was in the essential amino acids. The flies were not interested in the carbohydrate, including complex carbohydrates. They weren't interested in lipids, nor were they interested in the non-essential amino acids, which are the same in flies as they are in mammals. It was only the essential amino acids in the diet that were important. And to summarize a long series of experiments, first of all, it turned out that methionine alone was sufficient and necessary for fecundity. So if we give back the flies methionine, their fecundity is fully restored to fully fed levels, and their lifespan is not affected at all. So what we then have is a long-lived, fully egg-laying fly. So there's no trade-off between those two traits. They're independently controlled. And if we give back everything except methionine, there's no response. And we think that what is going on here is that from the fly's point of view, the yeast is deficient in methionine compared with what the fly needs to produce eggs. I can go into that more at the end if anybody's interested. But we can completely uncouple the fecundity and the lifespan responses. And it turns out that some combination of the essential amino acids, and particularly imbalance between them, is what's important. So what's killing the flies when they're on the high diet is either having to put up with the presence of or having to get rid of essential amino acids that can't be used in reproduction or personal maintenance. And it also turns out that if we take flies that are one of those long-lived insulin signaling mutants that I've shown you, it blocks both responses. Their fecundity and their lifespan don't respond. So these flies need an intact insulin signaling pathway to respond to these amino acids. We're now actually starting to do very similar experiments with mice in Cologne, but it's already very clear from the literature that health during aging in mice depends very much on macronutrient balance in the diet. So if you give a mouse a diet that doesn't have enough protein in it, it has a protein target, and it will overeat the other components of the diet to meet that target. And also high protein levels are deleterious. And it's also very clear that some amino acids are more important than others in mice. So I really think in most organisms this is going to turn out to be dietary restriction, not calorie restriction. And if we understood the responses better, I think it could be useful for human health. I'd like to say a few words now about aging-related disease and the relationship between this work on aging and aging-related disease. We've seen that if we reduce insulin signaling, that we can improve the natural health of the animal during aging. There are also an increasing number of studies now looking at the crossover between these interventions that slow the aging process and specific genetic models of aging-related disease. And I'll just talk a bit about Alzheimer's, but very similar work has been done on cardiovascular disease and on cancer models as well. So I think I don't need to point this out for this audience. Alzheimer's disease is the most common form of dementia associated with these two pathologies, the extracellular amyloid plaques, the intracellular neurofibrillary tangles from hyperphosphorylated tau. And from our point of view, it's an interesting one because the vast majority of cases are sporadic, with age as the main risk factor. So what we wanted to do is to use the flies to ask about crossover between this type of pathology and the interventions that ameliorate aging. And the work was done by this group, 3 postdocs, Onyx, Ian, and Fiona Kerr, and Ian, who was a graduate student in the lab at the time. Some of you may not know that flies are very frequently used as a model of neurodegenerative disease. And the reason is this. The compound eye of the fly, which should be there, but for some reason isn't. So what one can do is to express, using a gene expression system, in the eye of the fly as it develops, toxic peptides associated with particular neurodegenerative diseases, so in this case, A-beta or tau. And that beautiful arrangement of mosaic ommatidia in the eyes is very easily disrupted by the expression of toxic proteins during development, and that can be used to screen, both for toxicity of proteins and for things that rescue that toxicity. But we wanted to modify this system somewhat so that we could study what went on in the adult fly, because we were interested in the aging-related aspect. So what we've done is to modify a type of fly that was actually produced in Cambridge, particularly through the work of Damien Crowther, where A-beta 1-42 Arctic, one of the toxic human familial types of A-beta, is produced in the neurons of the fly, but only once it becomes adult. So what we use is this gene switch system. And depending what promoter you put in this case LV, this makes it expressed in neurons. And when the product of the driver is produced, normally it would bind to the upstream activating sequence and drive the expression of whatever's downstream. But in the gene switch system, this is modified so that when the two come together, this doesn't necessarily happen. If the inducer, which is called RU486, which is a steroid hormone, isn't there, if it's not fed to the fly, this is not capable of driving the upstream activating sequence. But if we feed them RU once they've become adult, then this binds to the modified GAL4, and now we get expression of the transgene. So what we've got here is a system where we can drive expression of A-beta 1-42 Arctic in the neurons of the adult flies. That's just shown here. This is what happens at the messenger RNA level. The expression actually goes down slightly with age because the flies have to eat the inducer, and older flies don't eat so much. But the protein accumulates, and it accumulates in a mainly insoluble form. So this peptide actually has the export sequence on the front of it, but it looks as though in flies there's a mixture of intracellular and extracellular A-beta. So what we've done with this model is, first of all, to characterize the phenotypes associated with it. And very clearly, the expression of the Arctic peptide in the brain shortens the lifespan of the flies. It also produces electrophysiological defects. So you can do electrophysiology with flies using this giant fiber escape system. And this is what mediates the visually mediated escape responses. It's a very large interneuron which travels posteriorly in the fly, and synapses either with other interneurons or directly with motor neurons, and then activates these two sets of muscles. So this tergotrochanteral muscle is the jump muscle. So the first thing the fly does is to jump, and then this dorsal longitudinal muscle is the indirect flight muscle, so it takes off and escapes. And we can look at the function of this system by stimulating at the front end and then recording the arrival of the impulses in the muscles. And there are two things that one can look at here. The first of all is the response latency of the whole system. And we've got here flies expressing the peptide or not. And you can see that when they're young, there's not much effect on the latency. But by the time the flies are old, it's considerably increased in both sets of muscles. And this is actually also a natural aging-related phenotype of the flies. The transmission through the system gets slower and slower. And it turns out that the reason for that is the loss of electrical synapses. They don't lose neurons, but they do lose synapses. We can also look at how good the system is at keeping up with a rapidly fired sequence of pulses. So we've got different frequencies here. This is not an aging-related phenotype. It doesn't decline with age. But you can see that already in the young flies here, there's a deficit in their ability to respond to this train of pulses, especially at higher frequencies. And by the time they're old, that's become very highly statistically significant. So electrophysiological defects, also a behavioral defect. So one of the very obvious things about flies is they like to go up. And one can test how good they are at going up simply by banging down the tube in which they're present. So they all go to the bottom and then look how good they are at climbing up again. And I hope we'll see Onyx do this now. Yes. So these are the control flies on the left and the ones with the peptide on the right. One naughty fly stayed at the top. And you can see that these flies are about 15 days old. The wild types are very much better than the mutants at climbing. And we can quantify that. That's shown here. Again, this is a natural aging-related phenotype in flies. They get less good at climbing as they get older, but that effect is very much exacerbated by the presence of the effect of the toxic peptide. So, of course, what we've been doing now is to use this system to try and find things that can ameliorate these phenotypes. One interesting one that I don't have time to talk about is tau. So there's a very clear ortholog of mammalian tau in flies. And if we remove the tau, the flies are perfectly viable, as of course the mice are, but we greatly ameliorate these phenotypes. In fact, we completely lose this climbing phenotype and rescue a lot of the lifespan. So it looks as though something very like the amyloid cascade is going on in the flies. But as far as the insulin pathway goes, the main one that we've explored so far is, I guess, very familiar to many people, which is glycogen synthase kinase, which is, of course, very strongly implicated in human Alzheimer's disease. So the biochemical actions inhibited by insulin to promote glycogen synthesis, amongst many other things. Its levels are increased in the blood cells of Alzheimer's disease patients. It's required for the generation of the A-beta peptide from the amyloid precursor protein. Flies do have an ortholog of the amyloid precursor protein, but it doesn't cleave to give a toxic peptide. So actually this is not relevant to the fly system at all because we're expressing the peptide directly. And it's regulated by A-beta, which prevents phosphorylation of GSK3 at the serine 9, and that's an inhibitory phosphorylation, so GSK3 activity increases. Phosphorylates tau, and there's a fly ortholog, which is called Shaggy. So one of the things we wanted to know was whether if we inhibited the activity of GSK3, we might be able to rescue the pathology in this model. And we found very much that we could. We did it in two different ways, one of which is genetically, using a dominant negative version of GSK3 expressed in the fly brain, and the other was using lithium, which is, of course, a GSK3 inhibitor. I'll just show you the results for the climbing. So we've already seen this deficit in climbing in the mutant flies. If we put in the dominant negative GSK3 mutant, we rescue it completely. And the same with lithium, shown over on the right, the same rescue. Because deletion of GSK3 is lethal in flies, we're not absolutely certain yet whether this lithium effect is going through GSK3. But what's very clear is what both of them seem to be doing is inhibiting protein synthesis. We don't know the exact mechanism of that at the moment, but there's a very clear story developing in the context of amelioration of aging-related disease that knocking down protein synthesis can be very beneficial. There's also been quite a bit of mouse work on this kind of thing, and I think this is one of the clearest examples, which was actually worked out in the lab of one of my colleagues, Andy Dillon, in the Salk Institute, with the work led particularly by Ehud Cohen, who was a postdoc there at the time. And what they've done in this study is something rather similar, looking at the crossover, and they've used the mouse with the ubiquitous heterozygosity for the null mutation in the IGF-1 receptor, the Holzenberger, long-lived mouse. And what they have done is to combine that with a particular mammalian mouse model of Alzheimer's disease, which expresses both a mutant presenilin and a mutant form of APP. What they've asked, obviously, here with these crosses is whether introducing heterozygous loss of one IGF-1 allele can rescue the phenotypes in the AD model. So one of the very obvious things that this model does is to produce a memory impairment along with a number of other things. So just to produce a visual illustration of the work on the memory impairment. This is a Morris water maze, which I guess is familiar to quite a lot of people. And it's a test of the ability of the mouse to form and retain a spatial memory. So the way that it works is there's this opaque fluid, and you can just see a shadow in both tanks here, which is a platform, which is just below the surface. And initially what happens is that the mouse is introduced and the platform's above the surface. And it swims to the, or usually swims to the platform, because mice don't like being in water. Some mice actually roll over on their backs and refuse to cooperate, but most of them will swim to the platform. And in the course of doing that, they learn to associate the position of the platform with these external landmarks. And then in the test series, the platform's taken below the surface, and you ask the mouse whether it can remember where it is, because it's high enough that if it finds it, it can stand on it. And this is what the thing actually looks like. So this is the poor old Alzheimer's disease mouse who's been trained. And what you will see is that he swims more or less at random around the tank. He doesn't restrict his search at all in the area of the platform. Whereas if we take the rescued mouse, so this is the AD with loss of one IGF-1 receptor, he knows where he's looking, and he very quickly finds the platform. There he goes. So obviously that's just an anecdote. If one quantifies that, it's shown here. So these are the trials after the mice have been trained, and you can see that the deficit in the Alzheimer's mice, this is latency. They take much longer to find the platform compared with the wild types. Simply losing the IGF-1 receptor on its own doesn't have much effect on performance, but what it does spectacularly is to rescue the deficit in the Alzheimer's disease mouse, which is shown in the double here, the green line. So again, a very nice rescue. And in fact, they went on in this model to show that almost all of the phenotypes associated with this Alzheimer's disease mouse were rescued. This just illustrates the neuroinflammation down here with astrocytosis, but all of these other characteristics were rescued as well. So the main interest in this area now, and one to which a lot of people are trying to focus their attention, is actually to manipulate some of these targets pharmacologically to try to move towards development of drugs that might have some of these same effects. And one of the main successes in the area, again rather a recent one, has actually been with a different part of this signalling network, which I haven't yet mentioned, which is the target of rapamycin part of the pathway. So this is also in a nutrient sensing network with the insulin IGF network, and amongst other things, it senses amino acids. It's one of its main biochemical functions. And there's a drug called rapamycin, which not surprisingly inhibits the target of rapamycin kinase, because that's how it got its name. And it's a drug actually that's approved for human use. It's used as an immune suppressant, as a cancer chemotherapeutic, and I think to prevent restenosis in coronary artery disease. So it's a licensed drug. And very recently, it's turned out to extend lifespan in mice. So this study comes from the National Institute of Aging intervention testing program in the US. So what they do is to have an annual competition for any chemical that people want to introduce. And if the idea is accepted, then the chemical is tested in 3 different centers in the US. So these mice are the pooled results from all 3 centers. And you can see they started feeding the drug when the mice were already middle-aged, and they get quite a nice extension of lifespan, which has actually since been replicated. And these mice, of course, now are being extremely thoroughly phenotyped. And again, they're starting to show a quite broad spectrum improvement in health during aging. And I do know some individuals, again, one tends to meet them at conferences, who are starting to take rapamycin themselves. But I'm not sure that, seriously, that this is something that one would really want to foster at all at this point. I mean, rapamycin has very clear side effects. It suppresses wound healing. It's an immune suppressant, as we've seen. And the people who are doing these mouse studies are starting to look very seriously for side effects. And they found that the incidence of cataract in the mice is actually increased in the presence of rapamycin. And also, very unfortunately, they tend to develop shriveled testicles. So I think something that one would hesitate to adopt immediately. But on the other hand, rapamycin is turning out to have quite broad spectrum and surprisingly so effects. And I've just illustrated some of the studies here. This one, to summarise briefly, is an immunisation against influenza in mice. So if you infect mice with flu when they're young and you don't immunise them, they just die. And at the dose and with the system that they're using here. And if you immunise old mice, they die almost as badly. There are a few survivors, but they go down very heavily. The young immunised mice are up here. But it turns out that if you pre-treat these mice with rapamycin before the immunisation, then they go up and join the young mice. And this seems to be an effect on the hematopoietic stem cells of the mouse. The exact mechanism wasn't worked out in this study, but it's an interesting additional effect of rapamycin. It's also been shown, partly as a result of a lot of work in Cambridge, that rapamycin can be beneficial in many models of neurodegenerative disease. I've just shown one here with amyloid beta levels, but this has led to a very interesting and thoughtful review, just talking about how the therapeutic scope for rapamycin is being extended. And of course, this is what you'd expect if it's slowing the aging process, and thereby reducing the risk factor for aging-related disease. I also think that TOR kinase may not be the best target in this bit of the network, and there are actually some promising downstream effects that one might go for. So one of the activities of the TOR kinase is to phosphorylate the two S6 kinases that are present in mammals. And it's recently turned out that if we knock out both copies of the S6 kinase 1 in the mouse, again, this is from our London consortium, Dominic Withers and Colin Selman, we see an extension of lifespan. And this may well occur with a lot fewer side effects than with rapamycin. So again, these mice have a broad spectrum improvement in health during aging, and actually the gene expression profiles in their livers look very like those of dietarily restricted mice. This is obviously a potential drug target, and indeed there are actually available drugs against the S6 kinase 1, which I think should be investigated. Again, one of the nice things about flies is that if we get a drug effect like that, we can try and figure out the mechanisms by which it's operating rather quickly. So this is a study that was led by Ivana Bedoff in our lab in London, and she simply looked at rapamycin in flies and found that you can extend lifespan in flies with rapamycin as well. It's not a very big effect, but it's a very highly repeatable one. And of course, then what you can do reasonably quickly in flies is to say, well, what happens if we make the flies mutant? What has to be intact in order for the response to rapamycin to occur? And what she found was that indeed, the activity of the S6 kinase, which we've just seen in the mouse, does have to go down. So if she puts in a constitutively active S6 kinase and gives them rapamycin, she no longer sees the extension of lifespan. And it also requires autophagy. So if she puts in a mutant that prevents the increase in autophagy that occurs when you give the flies rapamycin, again, she doesn't see the extension. So it looks as though the cellular cleanup process is important. But I think one of the things that we may well see is the sort of thing that we're seeing with rapamycin, which is an extension of therapeutic range for those kinds of drugs that go for this pathway. So one very interesting one here is metformin. Metformin, of course, now is the first line of defense against type 2 diabetes, and probably one of its principal targets is the AMP kinase. But a recent study in Dundee has shown that new users of metformin are also at lower risk of various cancers. None of the individual cancers were statistically significant, but the overall cancer incidence was definitely lowered. And of course, we've all seen the recent work with aspirin, showing that not only is it effective against cardiovascular disease, but now is turning out to quite considerably reduce the incidence of cancer, again, possibly through targets that are in this nutrient sensing network. So to wrap up, I think the message that's coming out of this, or starting to come out of the animal studies, is that if we want to protect against aging-related disease, then we have the possibility of protecting against the aging process itself. And to somewhat parody what's going on at the moment with the aging-related diseases, they do tend to be studied somewhat in isolation from each other in different research institutes, by different communities who go to different conferences and read different journals, and that translates into the different medical specialities. And I'm sure this will continue to be an extraordinarily successful agenda, as it has been. But I do think there's another way of looking at it, which is that we may be able to treat the underlying aging process itself in order to produce some broad spectrum protection against all of these aging-related diseases. But of course, there are going to be huge practical challenges in doing that, many of them scientific. So if we want to make people healthier as they age, one thing we don't know yet, even with the animal studies, is when we have to give the drug. We know very little about the timing of these effects. So it may be fine to start the treatment late in life. It may be that it has to start in middle age or even earlier. We don't know if these effects are cumulative or acute, and that's something that needs quite a bit of experimental work. The other thing is that quite a lot of bioinformatic modelling is being done on this network now, and it's beginning to look as though actually suppressing the activity of more than one point in the network may produce the maximum benefit, partly to prevent feedbacks within the network itself. And if so, that's going to require more than one chemical present in any protective medicine. Obviously, for any drug that a lot of people are going to take potentially for a long time, safety is going to be a huge issue. Doing clinical trials for an outcome of a general improvement in health during aging is not going to happen. It's not a recognised outcome of a clinical trial. So we may well be looking at starting with specific diseases and then extending the range, or extending the range of existing drugs, of course. The pharmaceutical industry, I think, just are very interested in this now. I find myself being asked often to go and talk to them about this. Regulators, of course, are going to play a major role in allowing clinical trials, and funding's always a problem. So, to summarise, aging's malleable and can be ameliorated by single gene mutations. This nutrient sensing network seems to be really important, and it's increasingly seeing that slowing aging by mutations or drugs, not always, but sometimes can protect against aging-related disease. And I think what we're looking at is the prospect of a broad spectrum preventative medicine for the diseases of aging. I very much hope so anyway. And finally, it's a great pleasure to show you my research group on the left in the Max Planck Institute in Cologne, mainly working on mice now. And this is the whole Institute of Healthy Aging in one pod on the London Eye last summer. And I'd like to thank you very much for your attention.